Hi everyone, welcome back. Today's focus will be on chapter 2, which is entitled The Chemistry of Life. Now, one of the reasons why we have to discuss a little bit of chemistry in our biology class is because chemistry will allow us to explain how we can look at molecules such as DNA, remember our genetic code? RNA, different proteins, different organelles like the nucleus, and we're able to explain that they're made out of smaller components, which, as you may recall from chapter 1, is one of the characteristics of life, the fact that there are emergent properties or hierarchical patterns. Within chemistry, we're able to explain that these molecules are made out of what we call elements, which are substances that can't be further broken down in. And if you've ever seen a periodic table, you have seen these elements on full display. And part of what we'll be discussing today is exactly what an element is composed of. What does it tell us? So when we talk about things like electrons, protons, neutrons, ionic bonds, covalent bonds, what exactly are we talking about? And how does that relate to things like proteins, carbohydrates, lipids, which are more aligned when we think about things that are from a biology aspect? So let's start off nice and slow, and let's talk about this thing called a bulk element. A bulk element, by definition, is anything that is required in large quantities. And for the human body, we have four of them that are considered bulk elements, and those are going to be carbon, hydrogen, oxygen, and nitrogen. And one of the things I want you to do is as we peruse through the chapter, I want you to take a look at the images, and you're going to notice that whenever they have a chemical structure on display, you're going to see a lot of C's, H, O, and N's, because that will be the symbol for our carbon, our hydrogen, our oxygen, and our nitrogen. Now, obviously, a periodic table will also tell us a lot more information. So we're only going to cover some of the basics because obviously we are in a biology class, not in a chemistry class. But do keep in mind that along the way, if you have any questions or concerns or you would like a better explanation, feel free to reach out. Now, within the element, there are atoms, and these atoms are basically divided into what we call protons, neutrons, and electrons. And part of what I would like to emphasize here is that each of these atoms will have a unique charge, and they will also be located within different spots in the element. So if you take a look at the first one, the proton, that one will have a positive charge and can be housed in what we call the nucleus area of the element. And that basically means the huddle. Think of it kind of like the center. Joining it in that nucleus area will also be neutrons. Neutrons have a neutral charge. And the last one are electrons. Electrons have a negative charge and they do not reside in the nucleus. Instead, they will be in orbit encircling the nucleus. And in fact, what we can do is we can take a look at our illustration that is below our slide, and we can see right here in the center, we have a cluster that's going to represent our nucleus. And as you can see right here, we have our protons with the positive charge and the neutrons with a neutral charge huddled together. And then in orbit, represented in purple right here, you can see your electrons. Now, the electrons have a negative charge, and the fact that they are in orbit, we're going to use that characteristics in a little bit to explain why the electrons will be able to combine with different elements to give us something that is more complex, such as a compound or a molecule. But before we do all that, let's talk about a few of the key terms that are going to help us in our journey of better understanding chemistry and how it's related to biology. And in order to do that, I want you to take a look at the image that's at the bottom of our PowerPoint. Right here, we have a little sample of what our periodic table tells us. And within this little sample, we see that we have a symbol, C, that stands for the carbon element. And beyond that, we see that there's a bunch of numbers that have been indicated for us. Now, the top number is going to tell us something called the atomic number. And what the atomic number tells us is how many protons an element has. And here's the thing to remember. The atomic number will always stay the same for the element, which means that the number of protons will always stay the same. So regardless of what periodic table you look at, carbon will always have a 6 because it will always have 6 protons. You cannot alter the number of protons for an element. That's going to stay nice and consistent. Now, if you compare the protons to the other atoms, so your electrons and your neutrons, what we see is that the electrons and the neutrons are able to switch their numbers per element. And in order to do that, we're going to have to use terms such as ion and isotope. An ion is when an atom starts to alter how many electrons it has. So anytime you see the mention of an ion, that means that what we're doing is we're either gaining or we're losing electrons. So that number can fluctuate. If you see the term isotope, that means that we are varying the number of neutrons that an element can carry. So whether you're gaining them or you're losing them. So for instance, carbon comes in different variations. You have C12, C14. Some of these isotopes are used for medical imaging. Some of them used by archaeologists, for fossil dating. They'll have different characteristics. They come in different forms. But the more important thing is to realize that these forms represent isotopes, and an isotope indicates that the element is altering or changing how many neutrons it has. So once again, the number of protons will stay consistent. That's represented by the atomic number. The number of the number of electrons can be changed, and that's called an ion. My apologies right there. And if you change your number of neutrons, that is then referred to as an isotope. The other key terms that you might see on the slide is mass number and atomic mass. So let's take a look at those two next. Mass number is going to indicate how many protons plus neutrons an element has. So if you know the number of protons, that means that you know its atomic number. And if you then also find out how many neutrons it has, by adding the atomic number plus the neutrons, you will then get the mass number. And atomic mass, right here at the bottom of the slide, that's going to indicate the average mass of all isotopes. So remember, isotopes are the different versions of the element that can be created by altering the number of neutrons. So if you were to calculate the mass of all the different versions and take the average of it, then you would have the atomic mass. And as you can see in our example, the atomic mass is indicated at the bottom of the box. Now, I do want to add a little disclaimer in that the setup of each periodic table can be slightly different, but overall what the periodic table will have in common is that they like to arrange their elements in numerical order based on their atomic number. So all of your periodic tables will start with 1, which is represented by hydrogen, and then they'll continue on increasing their atomic number and showing the element that is then represented by that particular number of protons. Now, since your electrons are in orbit, floating around the nucleus area, it allows them the perfect opportunity to interact with other electrons from elements and potentially form a bond. So we have to kind of discuss how they're going to be able to do that. And part of that is the fact that electrons carry amounts of energy as well as a negative charge. And electrons also like to be paired. They like to be summed up by 2s. So what we see happening is that electrons will often form what we call orbitals. And these are just energetic regions that will circulate around the nucleus area. And just a quick reminder, in the nucleus is where you find your protons and your neutrons. Now, an orbital, or that particular energy region, can hold at a maximum 2 electrons, so it can hold a pair. So then the question is, what if you have more electrons and you pair them up and they have the same energy level? How are you going to then showcase that if an orbital can only hold 2 or 1 pair? Well, in that case, you're going to form what's called an energy shell. An energy shell is multiple orbitals stacked on top of the same level because they have the same amount of energy. And what's important to keep in mind is that each orbital can hold a maximum of 2 electrons. So the number of orbitals will then indicate for you how many electrons can be held. So for instance, if I have an energy shell that has 2 orbitals, that would mean that at a maximum I can hold 2 times 2 is 4 electrons, because an orbital can only hold a pair or 2. Since your electrons are going to be the ones that are interacting from one element to the next, they then have the ability to form what we call a molecule, which means that you have at least 2 or more elements interacting with each other. Now, some of these interactions can be with the same element. In that case, it's called a diatomic molecule. So for instance, if we take 2 oxygen molecules and we bind them together and make O2, which is an example of the oxygen that we inhale, that inhalation of oxygen is called a diatomic molecule because it's 2 oxygens, 2 of the same elements. If you do different elements, you call it a compound. So probably the best example is good old-fashioned H2O. Anytime you have some water around or you drink some water, what you're drinking is H2O, which means you have 2 hydrogens that are interacting with an oxygen molecule. That would be an example of a compound because you have a variety of elements interacting. Now, the part of the element that will interact, or I should say more specifically, the part of the electrons that will interact is referenced to as the valence shell. So this is going to be the outermost shell. You know what? Let me show you this on an illustration. Let's go ahead and take a look at this illustration right here. Now, what you're looking at are called Bohr's models, and they're basically 2D representations of an element. So in the center, you notice that you have a little circle that's going to represent our nucleus, and that's where we're going to house our protons and our neutrons. And then surrounding the center, you have like these halos. These are going to represent our energy regions. Now, keep in mind that if the halo or the energy region only has 2, such as the first level right here, that would then mean that it's called an orbital, because an orbital can only hold 2. And the 2 are the 2 electrons that it's holding on to. Now, if you have pairs of electrons at the same energy level, then you have what we call an energy shell. So right here, the second layer, we can see we have 1 orbital here, here's a second one, here's a third one, and here's a fourth one. So the fact that we have multiple orbitals makes this an energy shell. Now, the energy shell, its goal is to be filled and be paired up as best as possible to make the molecule as stable as it can be. So as you can see right here, let me go ahead and erase some of these little features, makes it a little easier to see. As you can see right here, for instance, in my carbon, it turns out that our energy shell, which is the outermost shell, which is also called the valence shell, because that's the shell that we're interested in filling, has a few vacancies. The vacancies are indicated by the white circles. So you have 1 here, there's a second one, a third one, and a fourth one. So there are 4 vacancies to be filled, and these vacancies will be filled by electrons from other elements. So for instance, I can go over to my hydrogen right here and take a look at its valence shell, which is the outermost shell. And as you can see, hydrogen has 1 vacancy. So what I can do is I can bring my hydrogen molecule over here and get it to interact with this set of orbitals and fill it up. So the hydrogen and the carbon will join together. But I can also take another hydrogen molecule and bind it to the second spot, a third one over here, and then finally a fourth one over here. And when I do that, I am then creating CH4, which is also called methane. Now, as you can see, each element will have a specific number of vacancies. So nitrogen has 1, 2, 3 spots that it's trying to fill, and oxygen has 2 spots that it's trying to fill. Remember our H2O, our water? That means that I'm going to take my hydrogen molecule and I'm going to bind one on each side of my oxygen, creating my water molecule. Now, please keep in mind that when you're looking at your terminology, to know the difference between an orbital, an energy shell, and a valence shell. An orbital means that you simply have one pair of electrons that are at the same energy level. An energy shell means that you have multiple pair of orbitals that are on the same energy level. So in a Bohr model, they will be represented by this little halo right here, and you'll have multiple pairs that will be present. And depending on whichever the outermost ring is, that is then also going to be referenced to as a valence shell. So the valence shell doesn't tell you how many orbitals or how many electrons an element has. It simply points out which one is the outermost shell, which one you will be interacting with. Now, I should put my little disclaimer in here that not all elements are looking to fill their vacancies. Not all elements have vacancies to fill. So for instance, helium, which we use to, blow up our little balloons, helium is fully filled with electrons, so it does not bind with any other elements, and it's nice and stable. But in the case of our hydrogen, our carbon, our nitrogen, and the oxygen, you can see that they have all the opportunity for interactions to occur. Now, how exactly are we going to get them to interact with each other? We know we're going to be using electrons, but how are these electrons going to be mingling together? Well, part of it will be by forming different types of bond. The first bond I would like to discuss with you is called a covalent bond. A covalent bond is formed when electrons are shared between elements. And when they're sharing, it doesn't necessarily mean that they're sharing equally, because it turns out that each element has a different electronegativity. Electronegativity indicates the ability of an element like carbon, hydrogen, oxygen, or nitrogen to attract electrons. It is based on a scale, and you can take a look at the image right here below. It's based on a scale that runs from 0 to 4. The higher the number, the stronger the pull is. So for instance, let's say you have 20 electrons. If you are looking to do an interaction between oxygen and hydrogen, we can see that oxygen is stronger than hydrogen. So that means that if they're sharing the electrons, those 20 electrons, it's called covalent, but the oxygen will hold on to more, perhaps maybe like 14, while the hydrogen will hold on to 7 of them. So they're still sharing, but they're not equally sharing because one has a stronger electronegativity than the other. So what we're going to do is when you indicate a covalent bond, we will also have to indicate if one is stronger than the other or if they're relatively equal. Because as you can see from your scale, here in the category from 3 to 3.5, you see your nitrogen, your chloride, your oxygen. Those are relatively close to each other. So how are we going to indicate if there's like a strong and a weak pull versus a similar pull? Well, what we're doing is we're going to say it is either a nonpolar covalent bond or a polar covalent bond. A nonpolar covalent bond is going to indicate that the elements are relatively equal when we take a look at their electronegativity. So any interactions that would occur with elements that are relatively close by is going to be an example of a nonpolar covalent bond. So on your slide, it says equal pull, and an example of that would be diatomic molecule. Remember, those are the molecules where all the elements are the same. Remember our little oxygen example? So if they're equal, they're the same, then that would be an example of nonpolar covalent. They're sharing, and it's relatively equal sharing, 50-50. Now, on the opposite end, if you have one that is strong versus one that is weak, we're going to reference that as a polar covalent bond. Polar covalent bond means that you have one that is definitely strong and another one that is going to have a weak pull. So that means that when we're going back to our electronegativity scale, there will be one that will be more towards the higher end of the number scale and one that will be towards the middle or the lower end. Probably the best example of a polar covalent bond would be the interaction between oxygen and hydrogen, which we see happening in water. When water is formed, it is because the oxygen and the two hydrogens are sharing their electrons, but because the oxygen has a much higher electronegativity scale, it will hold on to more of the electrons than the hydrogens will. So covalent means we're sharing, nonpolar means we're sharing equally, polar means that one is stronger than the other. So you have an unequal sharing happening between the elements. Now, what if you don't want to share, right? What if you just want to take? Well, in that case, you're going to do what's called an ionic bond. In an ionic bond, what's happening is that you have an interaction where one side will lose electrons and the other side will gain electrons. And oftentimes when you deal with an ionic bond, you'll see the indication of a positive and a negative sign. And the reason that they like to indicate that is because if you recall, electrons have a negative charge. So if you're looking at an interaction between different elements and one of the elements is losing electrons, that would then mean that they are losing that negative charge and becoming overall more positive. The element that is then receiving these electrons is receiving all those negative charges and overall will then become more negative in its appearance. So an ionic bond is when you are going to head and you're doing an electrical attraction between two opposite charges. So one will receive and the other one will give away. There is no sharing. You're either losing or you are gaining electrons. I would say that the best example of an ionic bond is when you take a look at sodium chloride, NaCl, good old-fashioned table salt. So let's take a look at the Bohr model for sodium, Na, and for chloride. And I want us to take a look at the valence shell. Remember, that's the outermost shell. And if I look at my valence shell for sodium, what I'm going to end up seeing is that I have one electron. And remember, electrons love to be paired, so you have a solo electron, but notice how there's no vacancy spot. There's no little white circle next to that lonely electron. On the other hand, if I go over to my chloride, my Cl, what I see is that in their valence shell, it has a perfect vacancy spot that will just pair up that electron and fill up that orbital right here in its valence shell. So if I want sodium and chloride to interact and make table salt, I can't share because there's no vacancy on the sodium side. So instead what's going to happen is that sodium is going to give away its electron, thereby creating a valence shell that is full, and chloride will happily receive that electron, thereby also creating a valence shell that's nice and full. And what's going to happen to sodium? Well, it's going to become more positive because it gave away that negative electron. And chloride is going to become more negative because it received that electron. So as you can see on your illustrations, you have the indication of the positive and the negative side, indicating the fact that you have a swap in electrical charges. The other thing that I want to talk about when we do our little chemistry chapter is mention water. You know, I said to you before, water is H2O, and water is very unique in many different ways. And we know that our body is mostly made out of water. And we also use water as a basis for many things in science, including things like pH. One of the characteristics that water has that's unique is that it has both cohesion and adhesion. Cohesion means that water tends to stick together. Adhesion means that water tends to stick to other surfaces. And the way that water interacts is by the formation of hydrogen bonds. So cohesion, when water sticks to itself, can often be seen whenever you spill water on the surface. You're going to notice that it kind of bubbles up, Or as you can see here in this little example, you have a little insect that's able to hop on the surface of water. The reason for that is because the water molecules are forming a skin and interacting with each other. Or the next time you are in your kitchen, grab yourself a cup, turn on the faucet, let the water fill the cup, and let it go all the way to the rim of the cup. And you're going to notice right before it spills over, it's going to create this little bubble. That bubble right here is cohesion, water molecules sticking together. Adhesion is when we take a paper towel and we use it to dap up any water that we might have spilled on the surface or on ourselves. When you do that, the water soaks into the paper towel. That is then adhesion. Water forming hydrogen bonds with other substances. Or better yet, we can take a look at the illustration over here and we could see that we have a beautiful palm tree. And when it rains, this is my sound of making rain, water will fall on the sand, and then what's going to happen is the water molecules will start interacting with the plant cells, and by doing so, it's able to carry the water up the tree and throughout the branches and the leaves and everywhere that it's needed. And the water molecules will interact with the plant cells. That would then be an example of adhesion. So cohesion, water interacts with itself. Adhesion, water interacts with other molecules. And water can also be classified based on what we call hydrophilic versus hydrophobic. Hydro means water and philic means to love. So hydrophilic means that anything that can dissolve in water. This tends to be a polar molecule, so things like sugar, salt. Here's our little example of making salt water. So you can see that the salt is being poured inside of it. The salt represents what we call solutes, little particles. The water is the solvent, the liquid portion, and by combining it together, we create a solution. If you see that your particles do not dissolve, well, in that case, you might be looking at a hydrophobic solution. Hydrophobic means that it is water fearing. This tends to be a nonpolar molecule, and probably the best example of that would be anything that contains fats and oils. If you have ever tried to mix a fat, like a vegetable oil, and some water, you're going to notice that it forms two separate layers, and that is because it does not interact with each other. It's an example of a hydrophobic element or a hydrophobic solution. Water will also have different characteristics depending on what state it is in. So are you looking at water as a gas, as a liquid, or as a solid, like for instance in ice? And what we see happening is that those characteristics will depend on the hydrogen bonds that it forms. Now take a look at our illustration right here, and these little red and white dots, those represent our water molecules. The red is the oxygen, and the two little white dots, that's the hydrogen, so that's your little water molecule. And the way they're going to interact with each other is through this little black dotted line. Each dot represents a hydrogen bond, okay? So the hydrogen bonds form. If the hydrogen bonds are very dispersed and far away from each other, that's what water looks like when it's in a gas state. When the hydrogen bonds start interacting with each other in a very uncharacteristic, there's really not a pattern behind it, just kind of all over the place, that's what liquid water looks like. And then look at the interesting thing that happens. When you go from liquid to ice, so when water freezes and becomes solid, the hydrogen bonds will become extremely stable and very uniform in their shape. This allows the water to become less dense, and this is one of the reasons why whenever you add ice cubes to your drinks, it kind of floats to the top because it's less dense. And in nature, that's extremely helpful because if you look at our picture right here, we can see that we are looking at a lake in an extremely cold environment. And when it gets to a certain temperature, the water will freeze, and the frozen part of the water, since it's less dense, will go ahead and go to the top of the lake, creating a barrier that will protect the water below it from the extreme environmental factors that are happening above it. This is very beneficial to the organisms that live in the water because here's the deal. When water expands, when water freezes, it expands. So we don't want that to happen within the body of the organism, right? Because that could potentially be detrimental. So by shielding the environment, by creating that layer on top, it creates a mechanism for the water not to freeze over inside of the organism itself. Obviously, organisms will have other features like fur, blubber, extra adipose tissue that will also come into play to help protect them and keep them nice and warm and prevent the water from freezing too rapidly. We also like to use water as a basis for doing our pH analysis. And part of that indicates that when you look at a water molecule, when you look at H2O, it's determined, or I should say, it consists of a hydrogen and a hydroxide, an H versus an OH. And when we calculate the pH of a solution, what we're looking for is the ratio of hydrogen versus peroxide. And we're going to have three options, okay? Option number one is what we see in water, where everything is equal. So H equals OH, hydrogen equals hydroxide. When everything's equal, we call that a neutral solution. If the hydrogen exceeds the hydroxide, we call that an acid. And if the hydroxide, the OH, exceeds the hydrogen, we call that a base. So neutral, everything's equal. Acid, more hydrogens. Base, more hydroxides. Now, since we're doing a biology class, we're going to really talk more about pH in a numerical sense. So for that, we're going to have to take a look at the pH scale, which basically gives us a range from 0 to 14. Each step is going to represent a tenfold change. And the way it's going to work is, let me just draw my little line right here. So we start at 0, we're going to end at 14. The way it's going to work is that 7, which is right in the middle, that's going to represent neutral territory. So that means hydrogen equals hydroxide. Okay, so 7 is neutral. Any number that is less than 7 is going to represent an acid. So it's going to represent elements or solutions that have more hydrogens versus hydroxide. And the way it's going to work is that the lower the number, the more acidic. And that's because the lower the number, the more acidic. That means that you have more and more hydrogens and less hydroxides. Okay, so 7 is the middle. Any number that's below 7 is an acid. The lower the number, the more acidic. The lower the number, the more hydrogens, the less hydroxide. On the opposite end, any number that's above 7 is going to be considered a base or an alkaline solution. And in this case, it's going to go the higher the number, the more alkaline. So the more hydroxides, the less hydrogens it's going to have. And in case you're wondering, when we take a look at the human body, we're looking at a pH in our plasma or blood or our cerebral spinal fluid of around 7.35. Okay, so slight little range can come into there, but we're pretty neutral. Okay, so we're pretty much equal in hydrogens versus hydroxide. Here's a look at the pH scale that the book has provided, as well as some examples of solutions that you can find. Now, when you look at your pH scale, notice that they indicate for you how the hydrogen concentration and how it's going to go from high to low. And then it's also going to indicate for us the pH value. So that's our scale that's going to run from 0 to 14. And notice that right here by the 7, that's neutral territory, okay? And anything that is lower than 7 is going to be an acid. The lower the number, the more acidic. And anything above 7 is going to be a base or an alkaline solution. The higher the number, the more basic it's going to be. Also keep in mind that each step or each number represents a tenfold change. So if we're looking, for instance, at right over here, coffee, which has a pH of 5, and we're looking at soda or beer or wine, which has a pH of 3, we can say that this one is more acidic because it's a lower number. If I were to ask you how much more acidic, what you would do is you would see how many steps separates them, and each step would be a multiple of 10. So that would mean that it is 10 times 10 is 100 times more acidic. So cola, beer, wine, orange juice that have a pH of 3 is 100 times more acidic than coffee. If you were to do lemon juice, that would be 10 times 10 times 10 would be 1,000 times more acidic. So it works the same way for our bases. Each step represents a tenfold change or a log of 10, as many books will indicate. Alrighty, so pH is all about hydrogen versus hydroxide. If it's equal, it's neutral. If there's more hydrogens, it's an acid, which means the pH is below 7. The lower the number, the more acidic, the more hydrogens. If it has more hydroxide, it is a base or an alkaline solution. The higher the number, any number above 7, the more alkaline or basic it's going to be because it has more and more hydroxides. I'm sorry, it has more and more, yeah, it has more and more hydroxides. I said it correct. Our last part of our chapter is going to take a look at organic molecules. So now that we've kind of chatted a little bit about the carbons, the hydrogens, the oxygen, the nitrogens, we've talked about the ionic bonds and the covalent bonds. Let's talk about what our body likes to do with those individual pieces. Well, a lot of times it will take what is called a monomer, a little building block, a smaller unit, and it will link it together into a polymer. Mono means one, poly means many. So a monomer is your starting point, and the polymer will then be larger and more complex. And when we take a look at our body and we talk about polymers, we're often going to talk about things like carbohydrates, lipids or fats, proteins, and nucleic acids. Those are the four polymers that we are most familiar with. Also, what we're going to be doing is we're going to be using terms like dehydration and hydrolysis. Now, both of these terms will indicate the use of water, and that's because water will be essential whenever we're either trying to build or we're trying to destroy or pull apart. Let's go ahead and take a look at the illustration for dehydration and hydrolysis. Now, when you hear of a dehydration synthesis, I want you to think about the fact that we are building. So we are going from small to large. So in our case, we're going to go from monomer to polymer. And the reason they call it dehydration is because if you have two monomers and you're trying to link them together, oftentimes what you need to do is you need to create an available bond. And that bond can be created by taking a hydroxide on one side and a hydrogen on the other monomer and removing those two pieces. And then two things will happen. If I remove these, I will create a bond that I can then use to link the monomers together. So I've created something larger. And the second thing is that that hydroxide and the hydrogen that I removed, I can combine that and make water. So see in my reaction how I start with two separate pieces? I combine them and I lose water. Notice how this arrow is kicking out, meaning you're losing it. So since you're losing the water because it's what we call a byproduct or a waste product, we can then say that this is a dehydration synthesis. So I lost water, but I did it in order to build my monomer into a polymer. What if you want to go the opposite way? So what if you have monomers that are linked together? So you can see they're linked together right here, but I want to pull them apart. I want to go from something large to something small. So I want to go from polymer to monomer. Well, I'm going to use water again, but in this case, water is going to be inserted into the reaction. And when water is inserted, it's going to force the monomers to break apart. And what we're going to do is the water molecule will split into the hydroxide and go on one monomer, and it will split into the hydrogen and go on the second monomer. And the reason we need that is because when we pull the monomers apart, we have two empty slots. So we're going to replace that emptiness with either a hydroxide or a hydrogen. And the reason it's called hydrolysis is because hydro is for water and lysis is to pull apart. So you use water to pull everything apart. So dehydration means you're building. Hydrolysis means you are pulling apart, going from polymer to monomer. So let's take a look at how these will play in when we take a look at things like our carbohydrates. So carbohydrates are what we think of when we think about things like pasta, bread, potato, rice. Obviously, we're thinking about a lot of dietary intakes, but carbohydrates can be found in many different places and also have different uses. But what I wanted to display is that a carbohydrate is an example of a polymer. It's something that is large, something that's complex. Now, what makes up a carbohydrate? Well, that would be good old-fashioned sugar. And what you can do is you can use the term monosaccharide. Saccharide means sugar. Monosaccharide means one sugar. So an example of a monosaccharide would, for instance, be glucose. Glucose is what we call blood sugar. So when people say, I'm testing how much sugar I have, maybe they're trying to control their diabetes or whatnot, then what they're testing is how much glucose is in their bloodstream. How did the glucose get there? Well, most likely they consumed a carbohydrate. So maybe they were chewing on a bagel, they were eating some rice, and your digestive system then takes that polymer, that's the carbohydrate, and it does hydrolysis. It breaks it apart into smaller pieces until eventually it becomes a monosaccharide or a simple sugar. Along the way, we're going to talk a lot more about carbohydrates. So I definitely want to mention a few things. One of the things is that you can also see the term polysaccharide. A polysaccharide means that it is many sugars. So it is an example of a carbohydrate, just another way of saying it. And probably some of the most complex carbohydrates that we're going to encounter are going to be things like cellulose, chitin or chitin, as some people like to say, starch, and glycogen. Cellulose we're going to come across when we take a look at plant cell walls. Chitin you can find in the cell wall of fungi, so like mushrooms, but also in the exoskeleton of a lot of insects. So if you've ever like stepped on an ant or like a little spider and you hear that cracking sound, that's the chitin that is cracking in their exoskeleton. Starch and glycogen are more likely found as food storage units. So a carbohydrate is a complex sugar and its monomer is going to be a simple sugar or a monosaccharide. Another example of a polymer are proteins. And proteins we talk a lot about in biology. Proteins are composed of amino acids, that's their monomer. And when you look at an amino acid, it has a very peculiar structure, which I want to show you more about on the next slide. But what ends up happening is that when the amino acids link together, they're going to create a very special covalent bond called a peptide bond. So if you see any mention of peptides or polypeptides, then I want you to automatically think about the fact that you're looking at proteins, and proteins are formed by amino acids. Here is a look at some of the 20 amino acids drawn out for you. So here we have glycine, cysteine, and tryptophan. And as you notice at their structure, they have a lot of commonality, right? In fact, if we take a look at our little red box right here, we see a general amino acid structure. And what it's trying to show you is that all amino acids will have a central carbon, And that central carbon will make four connections or four sides. One of those sides is called an amino group, and that's because it has one nitrogen and two hydrogens. On the opposite end of the amino group is the carboxyl group, and that one has a carbon double bonded to an oxygen and a hydroxide. And the amino group and the carboxyl group will always be on opposite ends of each other. The other two sides will be dedicated to a hydrogen and what we call the R group. The R group is where the variety comes in. So depending on what structures lie in the R group will then give characteristics to the individual amino acid that you're looking for. Besides their differences in the R group, the amino acid will be exactly the same with the carbon in the middle, the amino group, the carboxyl group on its sides, and of course a hydrogen. Now, why do I make such a big deal about pointing this out? Well, because it turns out that when amino acids want to do dehydration and hydrolysis, they are very particular as to how they're going to go about forming their peptide bond. And it turns out what they like to do is that when they have two amino acids that want to link together, they're going to go over to a carboxyl group of one and remove the OH or the hydroxide from there. And on the other amino acid, they're going to go over to the amino group and remove the hydrogen. Very specific. So the OH comes from the carboxyl group and the hydrogen comes from the amino group. Once you remove the OH and the hydrogen from its specific location, it allows for the formation of the peptide bond, thereby creating a dipeptide or a larger molecule. And the same thing will happen when they do hydrolysis and they break that peptide bond. They will only replace the hydrogen on the amino group and only replace the carboxyl on the the hydroxide on the carboxyl group. So please make a little notation for yourself. It is very important to realize that when you're making a peptide bond, the hydrogen always comes from the amino group and the hydroxide always comes from the carboxyl group. One other notation that I would love to make about proteins, because we do talk about them so much, is that oftentimes you'll hear the saying structure equals function. A protein is very particular about how it will fold itself over to pertain to its ultimate function for the organism. If anything happens and the protein loses its structure, it will often also lose its function. When this happens, we call it denaturing. So anytime you see the term denaturing, I want you to think protein, and I want you to think the protein has lost its shape or its structure. And this tends to happen anytime there is an extreme alteration in its environment. So maybe too much heat, too acidic, too salty, anything that will upset its homeostasis or its regular environment. The example that you're looking at of the egg is probably one that many of us have experienced. If you take a raw egg and you crack it, the egg white is often translucent and clear. It is filled with this protein called albumin. When you apply heat to the egg, we're going to see that the translucentness becomes white and fluffy, and that is because the albumin starts to unfold, it starts to denature, it loses its function, which is fine by us because we're consuming the protein for its nutrition, not for its function. And as it loses its function and its shape, it gets a new appearance, hence the fluffiness of the eggs. We are disrupting the bonds. We are disrupting those peptide bonds that hold the amino acid together. Now, the third category for our polymers are going to be our nucleic acids. And our nucleic acids we're going to discuss in greater detail later on, but for now I just wanted to tell you those would be, for instance, when we look at DNA and RNA. DNA I've mentioned before in our previous chapter, that is our deoxyribonucleic acid, that's our genetic code. RNA is almost like its cousin. When we talk about transcription and translation, we're going to talk more about RNA. But for now, I would just like to show you that they are nucleic acids and they are composed of what we call nucleotides. Nucleotides are their monomers, and these are going to be the basis that you see illustrated right here. Notice that you see these little letters, A, C, G, U, and T. That stands for adenine, guanine, thymine, cytosine, and uracil. And the interplay of these nucleotides will determine the characteristics of the DNA and the RNA. Now, DNA is unique because it will have the T, the thymine, and RNA is unique because it will have the U for uracil. So T only belongs to DNA and U only belongs to RNA. The other letters, the A, the G, and the C, are found in both of these polymers. Our last example for this chapter, our last polymer for this chapter, will be our lipids or our fats. Now, lipids are not really a polymer. They fall in this really weird category where you have lots of variety within the lipids, so like your waxes or the lipids that we consume in our food intake. So what I would like to focus instead on is the fact that you have this subgroup in the lipids called triglycerides. And they are composed, their monomers are composed of fatty acids and glycerol. And based on their interplay or arrangement, these triglycerides can be classified as a saturated or an unsaturated fat. Now, a saturated fat means that its chemical structure is full of single bonds, which means that the fat is solid. So animal fats, bacon, butter, these are examples of saturated fat. If you like to cook and you go and you get yourself some raw meat, the white area that you usually see underneath the skin of the chicken or when you're doing your pork chops, all that white marble, that's all an example of saturated fat. Unsaturated fats are when we take a look at the chemical structure and we see a few double bonds. These double bonds are going to allow for kinks to form, which allow for the fat to be liquid. These are usually found in plant-derived fats, so olive oil is probably the best example. Most of us have bottles of olive oil that we've seen before. I have some in my pantry. You can just leave it out and that fat stays nice and liquidy. And that is because it is an unsaturated fat that has double bonds, so therefore tends to stay liquid. Now, please keep in mind that fats are what we call hydrophobic. They do not like to interact with water, but they're also energy rich. It will be very interesting to talk about that when we talk about the digestive properties of fats. But for now, what I would like you to think about is that if you were to compare a saturated and an unsaturated fat, if you had to decide which one was healthier, I would hope that many of us would be comfortable saying an unsaturated fat because an unsaturated fat, since it stays liquid, it's slightly easier for our body to process than the saturated fat, which tends to become solid. Of course, a lot of everything is dangerous, so please keep an eye on that. And then the last notation is this thing called a trans fat. What's that all about? Well, a trans fat is basically a bioengineered unsaturated fat. And the reason I say bioengineer is because some scientists decided that it would be best to remove some of these double bonds so that it could become a solid fat. And the thought process for that would be, for instance, for restaurants, it's easier to have solid units move around. So if you want to have a healthier fat, like an unsaturated fat that's liquid, if we can somehow turn it into a solid, it would make it easier for the whole process of going from point A to point B. So a trans fat is an unsaturated fat that has been manipulated to become solid. The problem with that is when you manipulate it, you make it just as bad as if it was a saturated fat from the beginning. So that's why some states in the US, as well as in Europe, are now banning the use of trans fats. So there you go. All right, so that is the end of our chapter number 2. I hope you enjoyed it. Keep in mind, if you have any questions, comments, or concern, please do let me know. And I will see you for the next chapter. We shall talk soon. Bye.